With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hi everyone, our question says given table gives an account of differences between PCR and gene cloning. Which of the following points show the incorrect difference? The first parameter is efficient. It says that PCR is more efficient and gene cloning is less efficient. So this point of difference is absolutely correct. So for PCR we have machines. So PCR is a automated process. It requires very less human effort as compared to gene cloning. So PCR is more efficient. So this point is correct. Now the second statement, it is based on apparatus requirement. So this says that PCR requires DNA whereas gene cloning requires restriction enzyme, ligase, vector and bacterial cells. So this point of difference is also correct. Now the third point of difference is based on manipulation. It says that PCR in vitro, gene cloning in vitro and in vivo. So let us first understand what is meant by in vitro and in vivo. So in vitro basically means that in vitro means lab, under lab conditions, artificially. So in vitro means lab. So PCR can be manipulated only in lab conditions, whereas gene cloning is a natural process which occurs inside organism. So it can be manipulated in lab as well as inside organism. That means in vivo. In vivo means inside an organism. So this point of difference is also correct. Now the fourth point of difference is based on cost and it says that PCR is more costly whereas gene cloning is less costly. So this point of difference is wrong. PCR is less costly because PCR has only 3 steps. So to perform 3 steps, we do not require much of a cost. So its cost is less. Whereas gene cloning, so gene cloning has a number of steps in and very complex steps. So we can say that gene cloning has many intricate steps to follow. So it is more costly as compared to PCR. So this point of difference is wrong. Now the fifth point is based on automation and it says that PCR yes, gene cloning no. So this is also correct as I told you that PCR is automated process. Sixth one is based on error probability and it says that PCR has less error chances whereas gene cloning has more. So this one is also correct. As gene cloning requires human effort, it has more chance of error. The seventh one is based on time. It says that PCR takes 2 to 4 days whereas gene cloning takes 4 hours. So this statement is wrong because PCR has just 3 steps, so it requires very less time. It requires 4 hours whereas gene cloning, it has intricate steps, so it requires 2 to 4 days. So the time here has been reversed. So this point of difference is wrong. Now the last point is based on application and it says that PCR has more application as compared to gene cloning. So this one is also correct. So we can see here that statement of point number 4 and point number 7 are wrong. So the correct answer for this question is option C, which says that 4th and 7th parameters are describes wrong differences, and all the other options, that is 1 and 3 is correct, so this option is wrong. 4, 5 and 6, so 5 and 6 are correct difference, so this statement is wrong. And D, 4, 7 and 8, so 7 and 8 are correct differences, so this option is also wrong. Hence the correct answer for this question is option C, that is point 4 and 7 states the incorrect difference. So this is the answer. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.